Hello, everybody, and welcome to the Monday Lunch live stream. Today, Professor Nick Huntington will be giving us a very exciting talk about targeting natural killer cells in cancer. My name is Paul Neeson. I'm from the Cancer Immunology program at the Peter MacCallum Cancer Centre, and it's my privilege today to co-host this session. I would like to acknowledge the traditional custodians of lands on which myself and today's presenters and audience are located. I would also like to pay respect to elders, both past and present, and extend that respect to other Indigenous Australians present online. So now onto the talk. So just briefly, Professor Huntington is the head of the Cancer Immunotherapy Laboratory at the Biomedicine Discovery Institute at Monash University. He's an international leader on natural killer cell biology with notable contributions to regulatory mechanisms of IL-15 signaling in NK cells, identification of human and murine NK cell differentiation pathways, novel xenograft, models to study human hematopoiesis, and identification of multiple checkpoints in NK cell activation and tumour immunity. Professor Huntington leads a research program aimed at deciphering the regulatory networks, the controlling NK cell immunity, and a drug discovery program using cutting-edge in vivo screens for novel checkpoints in NK cell activation for targeting cancer immunotherapy. He's president of the Melbourne Immunotherapy Network and is a co-founder of and chief scientific officer of Innate Proprietary Limited. I'll leave it over to Nick now for a very exciting talk, I'm sure. Thanks, Nick.
Yeah, so thanks for the opportunity. It's definitely been an interesting exciting couple of years in the natural killer cell space, and it's always thrilling to be able to, I guess, present natural killer cells to a non-expert audience and just bring into light their potential in cancer immunotherapy. So I'll go to just some disclosures. We obviously collaborate with industry and founded and started Innate, so we obviously are conflicted with natural killer cell therapies and exploiting them in cancer. So there's been obviously a large number of approved immunotherapy classes introduced over the last couple of decades, and a lot of them have been targeting towards T cells and have really given us unprecedented evidence that you can harness the adaptive immune system to have impact in immunotherapy. And those include targeted antibodies or immune checkpoint blockade or cancer vaccines and adoptive cell therapies, so transfer of chimeric antigen receptor T cells and also cytokines to boost some of these T cell responses to cancer. And they were some of the earliest immunotherapies that were approved. And they do all really, at the moment, focus on exploiting the adaptive immune response, so the T cell response, whether they be biospecific checkpoints or biologics, or the anti-CTLA-4 PD-1s that you're familiar with, the chimeric antigen receptors. Now we've just had another FDA approval yesterday or early in the weekend, late Friday in the US, for Juno BMS's CD19 CAR for cell therapy as well. And so this is, showing the potential of T cells, extremely amazing response rates in those lymphoma patients with CAR Ts, and similar data coming out with T cell engagers and biologics targeting tumor antigens and engaging T cells. And I guess the intracellular, some of the intracellular checkpoints there are less, let's say the proof of concept stage, less developed, but it's something that we're working towards in the field, is intracellular checkpoints in T cells as well as NK cells. But we've reached a point with combinational immunotherapy now that we've been able to improve on chemotherapy and monotherapy, and we've sort of reached this plateau now in really great response rates in some cancers, but they're a very small subset of cancers. And we've sort of reached the plateau now where we've, with the combination of the therapy, it might be hitting sort of 45% of patients in the best case scenario. And there is really still a great deal to improve upon to positively impact more patients by targeting the immune system. So we're looking at, what else can be brought into this question of combinational immunotherapy, additional cell types, additional drug targets, and, combining these rationally, not just, trying everything in combination. And we're running out of patients to do those trials, but really doing rational combinations of these immunotherapy trials. And also looking at different cell types. You can try to hit T cells 5 different ways, but is it going to really improve on the T cell response, or do you need to start to need bringing in accessory cells like the innate immune system? And that's basically what I really want to talk about today is the bringing the innate immune system more into play. So as I said, the current cancer immunotherapy approach is really T cell centric, but the innate system has the ability to recognize and kill cancer cells. It's a rapid first line of defense. It's not restricted through the MHC barrier, and it is really a bridge to drive and synergize with adaptive immune responses. So targeting endogenous immune cells is endogenous innate immune cells is predicted to increase the number of responders to immune checkpoint blockade and increase the potency of the T cell response and broader the indications for immune checkpoint blockade. So the area of interest for our lab and why we see the novel cell to sort of increase the number of responders in immune checkpoint blockade is natural killer cells. They definitely increase tumor inflammation and potentiate immune checkpoint blockade. So just a background is they are natural killer cells, the innate equivalent to CD8 T cells, which are the target of all of the real sort of successful immune checkpoint blockade and immunotherapy approaches to date. Natural killer cell development and function is dependent on IL-15 as is CD8 T cells, and I'll talk about IL-15 a bit later. NK cells are quite frequent in the blood, can be up to 20% in circulation, but also in spleen, liver. They're very frequent, up to that sort of 15 to 30% of mononuclear cells in these organs, and they're activated upon detection of altered self. So whether that be infection or transformation or DNA damage response, they've got activating receptors that can detect these alterations, but they've also got germline encoded receptors for MHC class 1, so they can detect changes or alterations in self, and that's just called missing self. And they use germline encoded receptors, not rearranged receptors like T cells, to detect cancer and altered self. The missing self, the presence and absence of MHC has been exploited therapeutically in hematological malignancies to induce some graft versus host effects in stem cell transplant, or graft versus leukemia in stem cell transplant. But really, there's still yet to have proof of concept of exploiting or drugging NK cells specifically in most cancers and definitely in solid cancers. So they're underappreciated in solid tumors. In metastatic melanoma, you can see they represent a very small proportion of the infiltrating cells, say 3% versus 50% of the T cells, for example. Yet 100% of these NK cells express activating receptors such as NKp46 and NKG2D, which bind and recognize melanoma ligands, whereas it's only a very minor proportion of those infiltrating T cells which are neoantigen specific. So in general, we'd have more melanoma-specific NK cells to CD8 T cells. And one of the receptors I mentioned, NKp46, it really plays a key role in detecting melanoma metastases in mice. And for example, when you remove NKp46, which is only expressed by NK cells, if you just remove that receptor, NK cell frequency is all essentially normal. You're just missing one receptor, but you now totally have lost your ability to detect and eradicate melanoma metastasis in vivo. So the impact of NK infiltration and activity in solid cancer outcomes wasn't really well documented. So a few years ago, Joe Kersons at WeHi, and now Director of Computational Biology at Innate, who by chance is also hiring at the moment for a role at Onco. So if anyone's interested in bioinformatics of cancer immunology, please reach out to Joe. But he designed and defined a gene signature looking at single cell data across healthy and blood samples, and also tumor samples from a number of melanoma patients. cross-checking that with genes expressed in tumors, but also genes expressed in T cells to try to get a unique NK signature. And he got this 20 gene signature where, you know, some patients have a very low NK signature and some patients have quite a high NK signature, and everything in between. So now if you bank those patients into orange, the high expressors versus the low expressors, you can see that the high expressors have a beneficial outcome over time, survival outcome in response to melanoma, and that's also true for breast and lung cancer and colorectal cancer as well. Recently, we've been digging a bit further into this, and Sebade in the lab has further refined this gene signature into sort of tissue resident and exhausted NK cells based on several recent RNA-seq data sets looking at healthy and colorectal samples, and then again cross-referencing them with T cells. And the reason for this is, you know, not all NK cells and T cells are created equal, and we see in tissues a large you know, accumulation and expression of sort of resident phenotype cells, so T cells and NK cells that aren't typically seen in circulation, but just reside in tissues. And if you look in tumors, there's a large sort of proportion of resident cells, and this is highlighted in orange here, versus the blue cells, which are the exhausted T cells and NK cells. And if you look across the different data sets, you can refine down to a very similar exhausted and resident signature across all the lymphocytes. And again, if you apply that to patient survival in colorectal cancer, it turns out that you know, having a high exhaustion and low residency phenotype is beneficial, and actually residency or NK residency looks to be detrimental to tumor immunity. And I don't, you know, whether it's the residency itself or the processes that drive the residency seem to be detrimental to tumor immunity, and I'll touch on that a bit later. So how do NK cells benefit or impact tumor immunity or solid tumor immunity? Mid last year, we published a review which really details this. It's quite an extensive review, so you can get into all the details by reading this article. But basically, we've generated this, or come up with this sort of 12-step cycle, which starts off really from NK cell recognition, and then tumor immunoediting, immune escape, sort of evasion of sort of tumor suppression, establishment of hot tumors, sort of driving T infiltration, T cell infiltration, promoting you know, tumor regression, and also immune checkpoint blockade responses. And I'll go through the whole cycle in some detail during this talk, but again, just reach back to the article to dig deeper into any of these processes. So the first step in the cancer NK cell sort of immunity cycle is really has to be the recognition, this sort of cancer NK cell cancer interactome. And what dictates really, you know, an NK cell response is the integration of all of these signals, whether they be stimulatory receptors interacting with ligands, which are, you know, germline encoded ligands, which could be upregulated upon stress or transformation. Also inhibitory ligands, which can also be upregulated and downregulated in response to transformation, and their associated inhibitory NK cell receptors, such as NKG2A. The KIRs, or the inhibitory receptors for classical MHC class 1. There's also, in the tumor microenvironment, pro-inflammatory or stimulatory cytokines, 12, IL-12, 18, IL-21, IL-15 being very important. And then suppressive factors, so prostaglandins, TGF-beta, activin, adenosine. So you've really got these four sort of inputs that really deciding whether NK cell will be activated and essentially degranulate or not, and it's stimulatory receptors, activating cytokines, inhibitory receptors, and suppressive factors. So one of the activating receptors which is of interest to the lab is NKp46, and mainly because, as I mentioned earlier, it has a really strong dominant role in recognition of tumor cells and killing, but we don't know what it's binding. There's several ligands proposed, but none of them are really a tumor-specific ligand, and some of the binding that's been reported is quite weak. So we're very interested in what that receptor is interacting with and how to exploit that therapeutically. And here's a bit of data on why we think this stimulatory receptor NKp46 is so important. So, for example, in a mouse, as I said, NCR1 is the gene that encodes NKp46. So when that's deleted by having 2 copies of GFP inserted into the gene, you have normal numbers of NK cells. And I think in blue here is the mutant mouse versus red, the wild type, but they have normal numbers of immature, mature 1, mature 2, the phenotype, frequency, everything is very normal. They just lack one receptor. And if you lack that one receptor, you, as I said, become very sensitive to B16 metastasis. You can't detect and eradicate and remove these tumors from circulation. The same is true with LWT1, a BRAF mutant melanoma model. And also, if you implant these BRAF mutant LWT1 melanoma models subcutaneously and look at the orthotopic growth, that's also greater in mice that lack one activating receptor on their NK cells. So it really hints to an important role for this receptor in recognizing something on melanoma cells, and then leading to either eradication, but more likely to activation and production of cytokines and chemokines, which drive tumor inflammation and effective T cell responses. The next step in the cancer immunity cycle really is, I guess, the editing and escape of certain clones. So obviously, when there's mutations occurring, these occur in certain cells. There's additional mutations within tumor cells, and this generates a heterogeneous pool of tumor cells to start with. Natural killer cells, as we've shown, can bind and kill tumor cells. And I guess based on the ligands that tumor cells are expressing and the number of receptors or types of receptors that NK cells are expressing, you start getting an editing of ligands. So therefore, cells that might be very immunogenic, having a lot of ligands for NKG2D or DNAM or 2B4 or LFA1 or NKP30, those cells expressing high levels of those ligands might be preferentially killed, leading to an accumulation of cells that have lower expression or edited by NK cells. And then the next step is really that these edited cells can start to secrete suppressive factors such as TGF-beta, or even start to cleave off some of their tumor ligands to be invisible to natural killer cells. And this leads to something termed NK cell escape or immune escape. And that will lead to an enrichment of cells that are, I guess, less immunogenic to natural killer cells. The next step in the cancer NK immunity cycle is establishment of this primary tumor outgrowth and generation of a suppressive tumor microenvironment. And we've been interested in what kind of suppressive factors could impact on NK function. So assuming that these suppressive factors are a key mechanism of suppressing NK cell reactivity. It's not so much loss of ligands, but it's actually the ability of these suppressive factors or tumors to generate these suppressive factors, or infiltrating immune cells to generate suppressive factors to impact on NK cell responses, which is sort of driving further tumor growth. And two factors that we've been interested in is TGF-beta and activin A. The receptors for these suppressive factors are quite high in immature cells and sort of reduce with maturation. But when NK cells bind these suppressive factors, you see an induction of SMAD2 and 3 phosphorylation. Just concentrate on green and I guess blue, which is the high activin, and sort of low TGF-beta in blue. So you get an induction of SMAD2/3 phosphorylation, and then a real drop-off in granzyme B production, which is a cytolytic granule, which is the serine protease, which is important for tumor cell death upon NK cell ligation. And especially TGF-beta, you see a big drop-off in granzyme B expression upon NK cells binding TGF-beta. We also see upregulation of residency markers on NK cells exposed to TGF and activin, and downregulation of key transcription factors such as EMS, which drive a real cytolytic sort of Th1 program within NK cells, and a reduction in proliferation. So these suppressive factors have a very broad and profound effect on NK cell viability, proliferation, and function. So exposure to them and the concentrations of them obviously are going to impact on tumor control. And we spent a bit of time trying to dissect the real role of these factors and how they might impact NK cells in vivo. And to do that, we generated some mice that were deficient for the receptor, the TGF-beta receptor, only on NK cells, and also a mouse that has constitutive active TGF-beta receptor only on NK cells. And we started looking in the mice, and it was very clear that if the mouse lacked TGF-beta receptor, they wouldn't generate these what we call tissue resident or resident, tissue resident or tumor resident types of NK cells. Whereas if they were constitutively signaling through the TGF-beta receptor, they would all generate or all become this resonant phenotype. So the TGF-beta signaling drives expression of a number of these residency markers in vivo, and it also does that in vitro as well. You can supply TGF-beta to NK cells and see that it upregulates DNAM and TRAIL, and also CD49A, which are associated with residency. So this immunosuppressive factors, drive an NK cell residency phenotype in tumors, but also broadly suppresses the function of NK cells, and some of that might be linked to residency directly, and therefore that impacts on the ability of NK cells to control those solid tumors. And this is really the key data, in vivo data, of the suppressive role of these factors. In a B16 F10 lung cancer model, I guess if you are resistant to TGF-beta, you have a lower or fewer metastasis in the lung, whereas if you're constitutively seeing TGF-beta, you have a very high level of metastasis in the lung. If we inject mice with small cell lung cancer models, and the TGF-beta receptor deficient NK cells are resistant to metastasis to the liver. And again, the TGF-beta receptor 2 mice, if you look inside the tumors, their NK cells all look like this kind of exhausted or conventional phenotype, whereas in a normal mouse, you start to see upregulation of CD49A and even downregulation of EMS, indicating that they're becoming resident in phenotype. And blocking TGF, as I said, has a profound effect in NK cell tumor immunity, but also in CD8 T cell function. And here's a melanoma model, for example, where we've deleted TGF-beta receptor from CD8 T cells in blue, and the mice are totally resistant to orthotopic tumor growth. And if you delete CD8 cells in green in those mice, then you become sensitive again to the tumor growth. So again, TGF is very dominant suppressive of NK and T cell function. And we've also done some experiments in sarcoma where we've blocked activin using follistatin in blue. And again, we can start to see some beneficial responses or suppression or regression of tumor growth when activin, this other suppressive factor, is blocked in vivo. So bottom line is that immunosuppression is a really important problem, important area of further investigation to try and understand therapeutic ways to overcome this in solid tumors. Now, throughout the whole cancer immunity cycle, we see that circulating tumor cells are extravasating and going into circulation. So metastases are extravasating, going into circulation and leading to metastases at distal sites. And NK cells are really essential to this sort of detection and rejection of experimental metastases. So, for example, is it going to work there? Okay, well, it's missing a panel here. But for example, if mice have no NK cells and you inject small cell lung cancers or B16 melanomas or EO771 triple negative breast cancers, whatever you like, if you inject these intravenously into the mice, normal mice can detect them and kill them in circulation quite efficiently, whereas NK cell deficient mice have no effect on resisting these metastases. It's very NK specific. CD8 deficient mice have normal control of metastases, so it's really NK cell specific phenomena. And that was all in experimental models where cells are injected intravenously, but we've done some spontaneous models, and here's a spontaneous model we did in collaboration with Thomas Gebhard, and this is all from Thomas Gebhard's lab at PDI, where he's developed an epicutaneous model of melanoma where B16s are just put on the skin, And a melanoma can develop and be progressive and result in metastasis, or it can be stable, or mice can be tumor-free. And Thomas published a great paper in Nature a couple of years ago now, defining this model and the role of TRMs in this sort of stable disease. But what we're interested in is like, what is, what's really controlling the metastasis? So the cells that are getting out of this epicutaneous melanoma and into draining lymph nodes. And to look at this, mice were inoculated with tumors. These tumors were surgically removed, and then we're looking at metastasis over time after the surgical removal of the primary tumor. And you can see control-treated mice have a small number of metastases in black in this pie chart. If you delete NK cells with NK 1.1 antibody, you have a lot more metastases, and also in the NK cell deficient mice, you have a lot more metastases as well. So this is a really nice model because it's spontaneous. It's a confined orthotopic tumor in really an epicutaneous space of the skin, and then the tumor cells are able to extravasate, and they're detected and eradicated by NK cells. And that's a similar situation when you have a very potent sort of transgene-driven breast cancer model, the MMTV POIMT model. We see that this is the driven, this results in a sort of a spontaneous breast tumor over time in mice, and we've crossed these mice to the NK deficient background and compared them to control mice. This is all on black 6 background, and basically the onset of tumors, the tumor growth sort of endpoint, didn't really change in the absence of NK cells. So NK cells weren't having a huge effect on this transgene-driven breast cancer model. But when we looked at the lungs of these mice, control mice, mice that have NK cells, we never saw metastases in the lung. However, NK cell deficient mice, we could very easily detect PYMT staining and metastases throughout the lung of the mice that lack NK cells. So again, NK cells can specifically prevent metastasis. Now these are even class 1 positive cells, so they're not detecting it based on the absence of class 1. These express high levels of MHC class 1, but they're detecting it based on expression of certain ligands on the tumor cell and eradicating it. And it does ask a really key question of, why does primary tumor onset and growth not change in the absence of NK cells, but the metastasis rate is very different? And how do primary versus metastasis, primary tumors versus metastatic tumor cells, how do they differ in terms of NK reactivity and suppression of NK cell function? So the next step, and this is probably the biggest, sort of the biggest revelation over the last 3 or 4 years in the sort of NK solid tumor space, is that NK cells are really important in potentiating immune checkpoint blockade outcomes by driving tumor inflammation. So we see strong correlation between conventional type 1 dendritic cells and NK cells, and T-cell dependent PD-1 responses in solid tumors. So example, in MC38 tumors, we look quite a number of different experiments, different colors here, but basically, we always see a correlation between NK cell frequency and CDC1 frequency on type 1 dendritic cells, and then sort of an inverse correlation with tumor weight and the frequency of dendritic cells and the frequency of NK cells. In this MC38 model is T-cell dependent and PD-1 responsive, but even just the role of endogenous NK cells, if we remove endogenous NK cells here in the open circles, tumor growth is enhanced compared to control mice. So this, is again telling us that NK cells are contributing to T-cell immunity in this model, which is a strong T-cell dependent model. And if you look at knockout mice versus wild type mice, the NK cell deficient mice have very few CDC1s in the tumor. And Keitana Risa-Souza and Kevin Barry and Max Krummel have discussed and described this phenomena also in mouse models. So it's gaining a lot of traction and definitely a lot of interest to understand how NK cells drive and activate dendritic cells in this process. The major recruitment of dendritic cells seems to be by this XCL1, XCL2 chemokine pathway, and it looks like, you know, FLT3 and potentially GM-CSF, which is made by NK cells, is also important for their sort of maturation and activity within the tumor. So moving on from that into sort of step 7 is that NK cells potentiate immune checkpoint blockade outcomes. So NK cells are required for optimal T cell function, and that is because they NK cells make a lot of RANTES or CCL5, and this is important for T cell, seems to be important for T cell recruitment. And also NK cells make a lot of TNF and interferon gamma, and this seems to help polarize T cell responses. So in black are just control mice, and in blue NK cell deficient mice, and you're looking at the number of T cell TNF positive CD8 T cells or interferon gamma positive CD8 T cells within the tumor. And you can see staining here for CD8 in brown in a wild type or control LWT1 tumor, whereas we don't see much or many CD8 T cells at all infiltrating into an NK cell deficient tumor. So sort of just to summarize that, NK cell infiltration within the developing tumor, we see NK cells infiltrating, and yet to fully understand the factors that drive NK cell infiltration. The chemokines, it's not simply you know, CXCR3, you know, CCL9-10 pathway axis that drives it. But NK cells do get into tumors and then react to tumor antigens, become activated. And potentially, if those tumors are making pro-inflammatory cytokines, that can also enhance their activity. But NK cells can kill tumor cells, generate tumor antigen. They also make chemokines to drive in conventional type 1 dendritic cells, which can take up tumor antigen to the draining lymph node, prime CD8 T cells, antigen-specific CD8 T cells, and they can also be recruited back in through the RANTES that's made by NK cells within the primary tumor. And there's a couple of really nice papers of late which have highlighted how NK cells really potentiate immune checkpoint blockade outcomes. So, you know, in the cycle, we see tumor regression in response to strong T cell control of neoantigen, but then again, tumors can resist T cells through upregulation of you know, PD-L1 or infiltrating monocytes, and other immune cells can also upregulate PD-L1 to suppress T cell responses. And thus, you know, is obviously clearly successful therapeutic concepts where you block this interaction between PD-1 on T cells and PD-L1 on tumor cells or other infiltrating cells, and this again can overcome resistance and lead to regression, or further regression or control of the tumor. So work from Rachel Zemek out in Western Australia, they developed a couple of models, and this one's an AB1 mesothelioma model, but they place the tumors on either side, so contralateral tumors, and they behave equally when you start to challenge the mice with immunotherapy. So CTLA-4 and PD-1 treatment at day 10, if you look at the left-hand side tumor and the right-hand side tumor, you know, some tumors don't respond and some tumors do respond, but they respond equally on either side of the mouse. So it's good. There's, you know, heterogeneous responses to immune checkpoint blockade, which is what we see in humans, but also you know, what we see in mice as well. And if you then, you know, remove one of the tumors just before you start the immunotherapy, you can actually sort of study what the microenvironment and infiltration looks like in responders versus non-responders. And Rachel's work revealed that responders have higher infiltration of NK cells and activated NK cells versus non-responders. You know, this was very you know, very clear in the RENCA, so this renal cell carcinoma model, but also in the AB1 model. And other groups have gone ahead and further confirmed that you know, the RENCA model is obviously very, this renal cell carcinoma model is very sensitive to PD-1 and CTLA-4 blockade. But in fact, the model or the response to PD-1 blockade is actually very dependent on NK cells. So if you remove NK cells, you no longer you know, have efficient control of the tumor. You no longer have this you know, anti-PD-1, anti-CTLA-4 effect if you don't have any NK cells. So it's definitely not working on the NK cells themselves, but it seems to be that NK cells are really there and required to drive better T cell infiltration and function, and therefore allow T cells to respond to immune checkpoint blockade effectively. So it confirms the role of NK cells in immune checkpoint blockade in mice, and really supports the combinatory approach for targeting natural killer cells in immune checkpoint blockade. Another model that is also very demonstrative of this process is when you block prostaglandin in tumors, so COX-1 or 2 knockout tumors, or prostaglandin PTGS knockout tumors. They have very low neutrophils and very high frequency of natural killer cells. And again, this is a very NK cell dependent model. And what you have is normally in a prostaglandin deficient tumor, in the white lines, the white dots here, it's well controlled. If you remove CD8 T cells in green, or even so NK cells in blue, you see that you remove the protection of these remove the protection of these mice to the tumors, and it really sort of hints that NK cells are important for a lot of the sort of pro-inflammatory factors and RANTES, even contributing to CXCL9 and 10 production within the tumor. But mostly it shows that in this model and in the presence of NK cells that you have potent T cell immunity. So if you're looking at these prostaglandin deficient tumors, you have big proportions, 42% of interferon gamma positive CD8 T cells. If you remove NK cells, you remove all of these interferon gamma positive T cells within that model. So that's, again, showing that NK cells potentiate immune checkpoint blockade in mice, and prostaglandin is a key factor in remodeling the tumor microenvironment and suppressing NK cell function. And similar data now is sort of filtering out amongst human data sets. And this is an example of melanoma patients on anti-PD-1 therapy. So basically they've banked the responders to PD-1 therapy and the non-responders in metastatic melanoma, and looked at sort of biomarkers of response, and they see that CDC1 and NK cells are really the only infiltrating sort of biomarker of whether patients have responded or not responded to immune checkpoint blockade. So that's, again, it's not Tregs, it's not CD8 T cell numbers, it was NKs and CDC1. So again, pointing towards the importance of these cell types in driving tumor inflammation, T cell infiltration, and immune checkpoint blockade responses. So I think that that's largely most of the immunity cycle. I'm not going to spend a lot of time on cold tumors and sort of immune checkpoint blockade non-responders, but I think that there is big potential to target these cancer types, or these subset of patients that are don't have infiltration or don't respond to immune checkpoint blockade. And I think that if we can target the tumor with biologics or chemotherapies, targeted therapies to try to ignite tumor inflammation, that's definitely a potential area of benefit. Trying to understand and exploit the reduced threshold for NK cell activation in some of these immune checkpoint blockade resistant patients, because a lot of the patients that are resistant to immune checkpoint blockade have mutations in MHC class 1. So to resist or evade T cell control, but in theory, if you lose MHC class 1, you become more sensitive to NK cell immune surveillance. So I guess understanding that pathway and potential avenues to enhance NK therapies in these immune checkpoint blockade resistant patients is also an area of keen investigation. And I guess looking at novel sort of DNA damage pathways and cell cycle pathways in cold tumors to improve NK cell recognition and immunotherapy. And we have a collaboration with a group in the States where we've got a couple of small molecules against these enzymes that are involved in DNA damage and involved in sort of tumor progression or sort of the tumor oncogenes. There's a couple of inhibitors that we have in combination, which result in a large upregulation of stress ligands on on cold tumors, and then that ignites NK cell infiltration and eventually responds to immune checkpoint blockade. And hopefully we can talk about that in a year or two. So it's a really exciting collaboration. Probably got 5 minutes. Just wanted to quickly skip over, novel NK cell drug targets. I've talked about IL-15 being very important for T cell and NK cell activation. So a while ago, we did a CRISPR screen looking for loss of what gene would make NK cells more sensitive to IL-15, and revealed that there's a SOX protein, CISH, which is induced by IL-15 signaling and encodes a sort of a negative regulator of IL-15 signaling, a protein which can bind to JAK1 and 3 and degrade the receptor to turn off the signaling. So a classic negative feedback loop in response to IL-15. And if we delete CISH or CIS in NK cells, mouse NK cells, or human NK cells, in red and blue are human CIS knockout NK cells. They proliferate a lot more, respond a lot more sensitive to IL-15 than parental NK cells. And again, mouse NK cells that are wild type for CIS or knockout for CIS. The knockout for CIS are obviously enhanced sensitivity to IL-15. It drives enhanced proliferation, survival, interferon gamma, granzyme, all of the classic IL-15 responses. So that's a negative regulator. It limits NK activation and tumor control. And we've done a lot of experiments in germline knockout mice where you inject metastasis or metastatic melanoma models, and the CIS deficient mice are resistant. And this is NK cell dependent because we can transfer CIS knockout NK cells into mice with no NK cells and also protect them from melanoma metastases. We've confirmed it using a conditional deletion of CIS in NK cells. And the mice are very, very healthy in the absence of CIS, germline deletion of CIS. So it looks like it's a pretty safe drug target. It's only expressed during sort of inflammation. So I think there's several companies will be interested in therapeutically targeting CISH in cancer immunology. But drugging it also in solid tumors, we start to see a very clear signal in CD8 T cells as well. So CIS knockout T cells proliferate more, they make more granzyme and interferon gamma in response to TCR signaling, as well as IL-15 signaling. And it turns out that that PLC gamma, which is a signaling intermediate downstream of T cell receptor signaling, is also negatively regulated by CIS. And then if we look at these epicutaneous B16 melanoma models that I mentioned earlier, which is sort of TRM mediated, the immunity, we see that CIS knockout are also protected compared to control mice and this is a T cell dependent model. Another T cell dependent model is a response to immune checkpoint blockade of these SM1 WT1 tumors, melanomas. There's no real monotherapy of PD1 blockade alone or CIS blockade alone, but we see clear synergy with CIS and anti-PD1 in combination. And also in MC38, a T cell dependent colorectal model that CIS deficient mice have better tumor control in the orthotopic setting compared to wild type mice. So again, CISH inhibition is synergizing with anti-PD1, and that would also be of clear interest to to biotech and pharma in expediting the development of inhibitors to CIS. But again, if CIS inhibitors are difficult to generate, which I'm sure they would be, there's a lot of other ways to drug CIS, and there's a recent paper from Nick Restifo's group looking at sort of the correlation between exhausted T cells in the melanoma, or melanoma resident exhausted T cells, and seeing that those that have sort of the higher level of exhaustion are sort of have sort of higher levels of CIS. And there was an idea so that CIS is inversely expressed with markers of exhausted phenotype. So CIS high in the sort of lower, exhausted cells. And there's now, there's probably 4 provisional patents by this group that's looking at CIS deletion in both NK cells and sort of CD8s for adoptive cell therapy. But basically, when they delete CISH in the patient's derived CD8 T cells, they see enhanced antigen-specific T cell immunity. You see greater interferon gamma and CD8 responses of CIS knockout cells against just general PMA inomycin. But also if you look in patient-derived CD8 T cells and stimulate them with the actual melanoma mutant antigen. So this GTF2E1, which is mutated in this patient, if you just take their normal T cells against this mutant, you get a mutant protein, you get interferon gamma and IL-2 and TNF production. But if you also delete CISH in those CD8 T cells, you get a huge amount of interferon, TNF and IL-2. So they were actually recruiting for a clinical trial a couple of years ago to look at CISH-deficient TILs or CISH knockout of TILs and then reintroduction. So take the TILs out, challenge them with tumor antigen, expand up the activated ones, delete CISH, and inject them back into the patient for sort of adoptive cell therapy. And they've stopped they've stopped recruiting for this, and I'm not sure exactly why. But the last thing I'll leave you with is part of the CISH phenotype is actually that CISH deletion partially overcomes some of the suppression of TGF-beta. So we noticed that CISH deficient NK cells, they have enhanced STAT5 activity, and they were still resistant to TGF in vitro. So wild type NK cells, when you treat them with TGF, you prevent their proliferation or dilution of CFSC on the x-axis. And TGF-beta knockout NK cells obviously resistant to TGF, but we see that the CISH deficient NK cells are still able to proliferate even at that high concentrations of TGF. And it wasn't complete, so we thought about this one mechanism of action of improved immunity could be resistance to TGF, but it's not complete. So what happens if we make double knockouts, delete CISH and the TGF-beta receptor? And we see some sort of enhanced effects over the single knockouts alone, and in fact in terms of like resistance to lung metastasis, and an ongoing experiment where MC38 control, it looks to be really well controlled in the CISH TGF-beta receptor 2 double deficient NK cells, kind of again points to combination immunotherapy of CISH and TGF-beta in solid tumors. I think I'm running out of time because of that hiccup at the start, so I probably won't discuss allo NK cell therapies. Just to say there's a lot of NK cell trials worldwide and a few CAR NK cell trials, and there's a huge number of benefits for using allo NK cells off the shelf to administer from any patient into a huge number of patients, as opposed to the very restricted and not safe, a lot of toxicity associated with CAR T cells. So there's a lot of benefit in looking at allo T and allo NK cells, and it looks there's a huge sort of shift towards allo CAR therapies, and that's probably one of the areas that NK cells are going to have the greatest impact in the short term, I think. So the points to take away: NK cells recognize melanoma via NKp46. The NK score of exhaustion greater than residency predicts patient cancer outcomes. NK cells make melanoma hot. They drive tumor inflammation. They prevent metastases. So cytokine checkpoints, CISH, that exists in tumor-resident NK cells and T cells. Suppressive factors, TGF and activin and prostaglandin are very suppressive to NKs and CD8s. And off-the-shelf allo cell therapy is the future for CAR therapy, in my mind. Check out the review for more details. We just think that Jim's obviously put T cell checkpoints on the map, and we're really hopeful to put NK cells up there, NK cell checkpoints up there in the next sort of decade as well. Last thing, the MIN 2021 Winter Symposium, so Melbourne Immunotherapy Network. It's back on for 2021. We missed it last year, but this is a great opportunity for face-to-face interaction up in Marysville. Here's the tentative lineup. A lot of great speakers across autoimmunity, cancer, infectious disease, even going to have a COVID-19 session. So please register, and it'll be great to see you all up there. And just thank everyone at Huntington Lab. I've put in bold the people who really contributed to the work, but Rebecca Del Conte, Fernando, Sebade, Hae-Young, the collaboration with Sandra Nicholson at WeHi, Thomas at PDI, and Jay and Joe Kirsons at Innate, and obviously all the funding. Thank you very much.
Thanks, Nick. That was excellent talk. So exciting, what's going on in the NK cell world. I'm delighted. So perhaps we can just start going through a few of the questions for you, Nick. In the chat session, Wendy Benson's asking, what about patients that have a large T-cell lymphoma? Is there any work to be done for them?
A large T lymphoma?
Yes.
Yeah. You know, I mean, in terms of the CAR space, I mean, there's obviously, you know, NK CARs against T-cell antigens is a, you know, and also allo is an emerging place as long as you can edit the T-cell antigen off LOTs. But you know, there's also been CAR T work against lymphoma. You know, therapeutically, there's not really any indication of you know, good NK cell targets or preclinical models for NK activity against T lymphomas at the moment. There's not a great deal of work done there, Wendy.
Yep. All right. Thanks, Nick. From the Q&A session, Robert Menzies is asking, my wife is currently on Opdivo to treat cholangiocarcinoma. Can she be given NK cell therapy simultaneously in either Melbourne or Brisbane?
Yeah, I mean, the short answer is no, but you know, this is really the aim, is to try to identify the best therapy, best NK sort of targeted therapy to elicit endogenous NK cell responses. And you know, at the moment, the clinical trials in sort of these biologics called engagers is, you know, really early stage, and also chimeric antigen or adoptive NK cell therapy also really in sort of safety proof of concept phase 1. So it's a little while to go, unfortunately, Robert, but you know, all the best for your wife, and wish you the best of luck.
So well, that's kind of a segue to a question I have. So you covered it really nicely towards the end. You talk about adoptive therapy with NK cells, whether it be NK CAR or others, allogeneic. Can you know, just try to provide a bit more detail about practicalities of applying that in the clinic?
Yeah, so I think, thanks, Paul. It's good. I would have loved to discuss it more, but I was conscious of trying to get enough questions. And so yeah, I mean, basically with the moment, autologous cell therapies aren't, you know, they aren't selling. You know, there's like 1,200 products got sold, I think, at least from, you know, maybe from Kite last year, and you know, they're not performing anywhere as sort of they expected. And part of it is, you know, the complexities of working with a sick patient's own T-cell product. And the logistics of harvesting them and sending them off, and you know, editing them and receiving them back and administering them into the patient. And then there's a lot of T-cell activity and cytokine release syndrome and neuropathy or ICANS, which means that most patients spend, you know, several weeks in hospital, you know, even weeks in ICU being closely monitored. So that's, you know, that's indirect costs and the direct costs of those therapies, but mostly the logistics and complications with manufacturing and just working with one product. With NK cells or even allo T, but with NK cells, you know, you can take any NK cell from any patient or any healthy donor around the world and grow them up and edit them and freeze them down and administer them to any patient. They can be all frozen and be used off the shelf because NK cells don't you know, participate in some of these CRS and ICANS. They don't undergo sort of graft versus host disease. They don't respond strong to, you know, they don't respond strong to missing self or to mismatched MHC, which T cells do. So they're safe. Now the question with safety is always, are they effective? You know, what's the, you know, are they efficacious? And typically, you know, safe things might not be as effective as something that's a bit more toxic, because the toxicity is driven by the actual response of the therapy. But, you know, the benefit is manufacturing can be standardized. You know how a batch of NK cells is going to perform because 20 patients have already been treated with that product before. You know, you can really standardize the product. You know, it'll bring down the cost because you're working with a pipeline of pooled blood products as opposed to individual patients each time. So you can really bring down the cost there. And definitely the indirect cost of, you can treat patients, and Katia Rezvani, who we collaborate with in MD Anderson, has treated patients as outpatients, right? You know, not, you know, just in the clinic, not in hospital. So the indirect costs of not having to have, you know, ICU and, you know, extensive follow-up and medical intervention across the following weeks of administration mean the product would be cheaper and cheaper to the, you know, healthcare provider. But the key question is, how effective are they? And that's still yet to be established. They're safe.
Yep. Thanks, Nick. All right. The next question is from Ranjit Sinha. And the question is, does NKPs, NCRs regulate expression of each other?
Do NCR receptors regulate expression of the other receptors? I don't think there's been, detailed analysis of that. I mean, activation of NK cells can alter expression of some of the NCRs. NKp44 is a clear one that comes up with activation. Typically cytokine-induced activation, I don't think there's a huge amount of evidence to say that that they regulate themselves or regulate each other. You do see them to be downregulated upon ligand interaction. So there is obviously post-translational modifications and internalization of receptors upon ligand binding, but I don't think so.
Okay. Next one's from Paul Beavers. He's great talk, Nick. Very interested by the NK, CDC1 and T cell axis. Any speculation on the regulation of XCL1 production by NK cells?
No, we see, in most of our experiments, we see all of the cytokine and chemokines being, going in the same direction, being regulated, as one. If you have a strong activation and upregulation of gamma, we tend to see, that the other cytokines, GM-CSF, XCL1, RANTES and everything going in the same direction. I don't necessarily have any ideas of a sort of specific regulation of XCL1, but it's something that Hae-Yong in the lab's really keen to dissect, at least in vitro, to try to understand what receptors or factors are mediating some of the XCL1, XCR1 crosstalk in tumors. Yeah. She's trying to look at that, at least in vitro, in a reductionist model of CRISPRing out certain receptors and factors. But no, there's nothing that I can really put my finger on as a key regulator of XCL1, Paul.
Okay. And the final questions are from Emma Petley. She's asking, have you looked at the memory effector exhaustion phenotype of CIS knockout T cells?
Phenotypically, it's steady state we have, and they look, really, relatively normal. I think there's an increase in tissue resident memory T cells in those mice in certain organs. But in steady state, those, the T cell subsets look relatively normal and are expressed at, normal frequencies. I'm just trying to think of the data, but I'm pretty sure there's an increase in TRMs in the skin and the liver. I think we did some experiments with Laura McKay and Simone Park on that a while ago, but
okay.
Yeah.
So the final question was from Emma was, can NK cells respond directly to anti-PD-1 treatment? In other words, do NK cells ever express PD-1?
Yeah, I mean, that's a bit controversial, I think, in the field, because there's, some mouse models where NK cells express a lot of PD-1. In humans, in vitro, in certain conditions, they can. There's evidence in solid tumors that NK cells do express PD-1. But I think most of the evidence suggests that the PD-1, PD-L1's not a dominant suppressive factor on NK cells. It's at least the therapeutic response is more T cell mediated. But it's quite controversial. I think at least there's a couple of groups in the US that sort of argue about this, and it might depend on the model used. But
yeah. So I think probably we should wrap it up there. There's a few more questions, but we're kind of well past 2 now. That was a great talk. It was so interesting, and it's on the link there that we provided at the start of the talk. Thanks so much, Nick, for making the time to talk to us and answer all those questions.
My pleasure.
And I think that winds it up for now, I think. Cynthia?
Yep. Hopefully see you all in Marysville 2021.
Yes, absolutely. Looking forward to it.
All right. Bye, Paul.
Okay. Bye.